Chapter 12, Neural Tissue. So Chapter 12 is the first of a series of chapters on the nervous system. And this one in particular is going to look closely at neural tissue. So we're going to start out by first reminding ourselves the main parts of the nervous system, and then we'll look a little bit more at the cellular level. So, as it says here, the nervous system includes all the neural tissue found in the body. And we can really divide that up into two major types of cells. We've got first our neurons, which are the cells that are actually going to send and receive signals. So if the brain is controlling a process, it will use neurons to send that signal to whatever organ or tissue we would like to act. Or if there is a sensory signal coming in that needs to be taken to the brain for interpretation, the neurons can bring those signals back to the brain. So it is a two-way street. Then we've got the neuroglia, which are also known as glial cells. These guys are the ones that will support and protect the neurons. And we'll take a close view at neurons as well as looking at the neuroglia, how they support and protect, as well as what kinds of neuroglia we will find. So before we go there, let's just take a quick look at a neuron, and we're going to start with a basic view and then we'll get into the details. So this picture is a pretty good representation of a typical neuron, though they do come in different shapes, different sort of looks. But we'll start with this type. So we've got this basic neuron, which is made up of a few main things. We've got first this area here, which is called the cell body. This makes up usually the largest area when we include these little fine projections, which are called dendrites. And we'll talk a little bit in a minute about what those do. But right about here, from this area down to here, we call this the axon. And then the axon ends in what are called synapses. And we can see all these little things that look like little buttons at the end. These are called the synapses. So we'll go into a little bit more detail on what each of those things do and also some of the other internal structures. Neuroglia, remember, was the other type of cell we'll find in the nervous system, and these are the ones that support the nervous system cells' neurons. And we can see here, this is a neuron. There's the cell body we just pointed out, and then down here we've got the axon. These guys here are examples of neuroglia, and we can see that they seem to be attached to the neurons, supporting them. Now this is just one function, but this just gives you an example of how the neuroglia are found surrounding and supporting the main cells of the nervous system, the neuron. So backing up a little, we know that there are two kinds of cells. We have the neurons and we have the neuroglia. But what about the basics of the nervous system if we kind of zoom out from that, looking at an organ level? The organs of the nervous system are the brain and spinal cord. The sensory receptors, these are the guys that pick up on sensations. And some examples would be things like the eyes and the ears. And then we have the nerves that connect the nervous system with other body systems. We can divide the nervous system into two main parts. We have the central nervous system, abbreviated as CNS, and the peripheral nervous system, abbreviated as PNS. Central nervous system is what it sounds like. This is the center of the nervous system, with the center axis of the body. Peripheral nervous system branches off of the central nervous system or to the periphery of. We're going to look at a picture of this to give you a visual. But before we do, let's look a little bit closer at the central nervous system, or CNS. the central nervous system, again, is made up of the brain and the spinal cord. Included in that, obviously, would be the neural tissue that makes up the brain and spinal cord, as well as connective tissues and blood vessels to feed and nourish the neural tissue. The main functions of the central nervous system are going to be to process and coordinate. And the kind of things that we're processing and coordinating are sensory bits of information from inside the body and outside the body. Motor commands, which are the actual tasks that the nervous system can direct. These motor commands can control activities of other organs in the body, like muscles, for example. And the higher functions of the brain, intelligence, memory, learning, and emotions, which we'll talk more about in a later chapter when we get into the details of the brain. The peripheral nervous system is going to include all the main neural tissue outside of that central nervous system that we just mentioned, so outside of the brain and the spinal cord. And the functions of it are going to be to deliver information to the central nervous system. So if we pick up on a sensation, the wind is blowing, somebody pinched you, those sensations will be carried back to the brain for processing so that we have awareness of what's going on. And they'll also carry commands out of the central nervous system to the organs and tissues if an action needs to take place. So again, it's a two-way street. The peripheral nervous system brings information in and helps carry directions out that are given to it by the brain or central nervous system. The peripheral nervous system is mainly made up of nerves, called peripheral nerves. Nerves are bundles of what are called axons. And we did look quickly at the axon at the beginning when we looked at that neuron. So nerves are made up of bundles of axons with connective tissues and blood vessels. Okay, and we're going to take a close-up of this as well. And their job, again, is to carry information and motor commands in the peripheral nervous system. And remember, we're carrying information into the central nervous system and out of the central nervous system. There are two categories of these nerves. We have cranial nerves, which are where they sound like they should be, connected to the brain, and then spinal nerves, which attach to the spinal cord. And we'll talk in more detail about the spinal cord in the next chapter that we'll be going over. Okay, so now we're going to begin with our visual here. So we've got the central nervous system and the peripheral nervous system. Again, remembering that central nervous system, meaning center, we have the brain and then the spinal cord coming down here. Now the brain and the spinal cord are no good if they can't direct the rest of the body's organs and tissues. So they need someone to carry that information in and out. And that's where the peripheral nervous system comes into play. So we've got the spinal cord here, and then coming out of the spinal cord, we see all of these nerves. Okay, so all of these guys here, all nerves. And those nerves are the peripheral nervous system. So central nervous system at the center, peripheral nervous system are the nerves that come out of the central nervous system or to the periphery. So we can divide up the peripheral nervous system functions into two major areas. We have the afferent division and we have the efferent division. The afferent division is the division that carries sensory information to the central nervous system from the PNS or peripheral nervous system receptors or nerves. And one of the ways that I remember this best is afferent starts with A, and A reminds me of accessing. Okay, so we're coming into or accessing the central nervous system. So remember that afferent, again, we're carrying information from other parts of the body to the brain. Okay, efferent division with an E carries motor commands out. Okay, so from the central nervous system to muscles, glands, or other tissues. So out of the central nervous system. And I remember this as well by remembering that efferent starts with E, and E also begins the word exiting. So to leave the central nervous system. So again, this is that two-way street thing. We have information coming in, afferent or accessing, and we have information going out to direct activities in the body, efferent or exiting. So some of the parts of the afferent division, remember this means coming in or accessing the central nervous system, are receptors. Receptors are going to pick up on any change or stimulus and bring that information in to the central nervous system for interpretation. And that kind of information can come from sensory organs like the eyes and the ears. So we see something, we hear something, that information goes through the afferent division of the peripheral nervous system to the brain, where we can then process the information and interpret it. Effectors are the ones that are going to respond to whatever information we pick up. So, for example, if our skin picks up that we are getting hot, this information will be carried to the brain, and the brain can then direct effectors to begin sweating so that we can maintain our normal homeostasis. So receptors pick up on a change, and effectors are going to respond to that change. Okay, so looking at the efferent division, we have the somatic nervous system, or SNS. There's a lot of abbreviations to remember here. And this part is going to actually control skeletal muscle contractions, voluntary and involuntary muscle contractions, or what we know of as our reflexes. Autonomic nervous system, or ANS, controls subconscious actions. So when our smooth muscle contracts, and remember when we say smooth muscle contracting, we're talking about areas like the digestive system or the uterus, obviously things that we don't consciously control the movement of. Cardiac muscle as well, good thing we don't have to be in control of our heartbeat, something we don't think about, subconscious. And the autonomic nervous system also is commonly divided up into two areas. We have the sympathetic division and the parasympathetic division. Sympathetic is the stimulating division. This is what we often refer to as our fight or flight nervous system. It typically is accompanied by, when it's stimulated, is accompanied by a rise in heart rate, blood sugar, and typically is a result of something that is either very exciting or frightening. The parasympathetic division, also referred to often as our rest and digest phase, has a very relaxing effect on the body. So this would be the opposite of what we just mentioned. Parasympathetic would be when we are terribly relaxed, and typically is accompanied by a lowered heart rate. Blood sugar is obviously not as high, and it's when we digest the best. More blood flow goes to the digestive tract, so that's where we get the rest and digest from. So sympathetic is again fight or flight, and parasympathetic, our rest and digest phase, our more calm and relaxed phase. So overall, autonomic subconscious, and somatic would be our skeletal muscle contractions that are voluntary and involuntary, like our reflexes. So going back to the neuron, now we know that we have two big divisions of the nervous system: central, brain and spinal cord, and then peripheral, which are, of course, the peripheral nerves that come off of the central nervous system. And both types, or both divisions of the nervous system, are made up of neurons. And The neuron, again, is the basic functional unit of the nervous system, and its job will be to direct all of the impulses that we mentioned before, whether they're coming in as sensory information or going out as motor commands to get something done. All of these actions are going to be carried back and forth through the neurons. So kind of imagine the neurons a little bit as your electrical wiring system, sending impulses back and forth between the brain, spinal cord, and the rest of the body. So the structure of a neuron, we need to understand that in order to figure out how they actually work impulse-wise. The multipolar neuron, this is the type of neuron we're going to focus on today, is a very common neuron found in the central nervous system. Okay, so right now we're focusing in on the neuron appearance in the central nervous system. And some of the main parts or anatomy of the neuron are going to start with the cell body, okay, which is the part we already pointed out. The cell body is covered with dendrites, which are the fine little branched structures. And then we have, coming out of the cell body, a long axon. And that axon is going to be carrying what we call our action potential, which we'll talk about a little bit later. So inside the cell body, what kind of stuff are we going to find in there? We're obviously going to find some of the main things that we would typically expect to find in a cell, beginning with the nucleus and nucleolus, which we know is going to house the DNA. The specialized cytoplasm for a neuron, which is called the perikaryon. Mitochondria, which produce energy. And then the rough ER and ribosomes. And if you recall, the rough ER, which is the type of endoplasmic reticulum that is covered with ribosomes, works well in producing protein. And specifically, the rough ER and ribosomes are going to be producing neurotransmitters. And neurotransmitters, recall, are chemical messengers used by the nervous system. So they are the messengers of the nervous system. They're going to be carrying information for us. And then the cytoskeleton, which is a network of proteins that give the cell shape and structure. Okay, so now that we've put all the verbal in, let's take a visual of this. Okay, so everything we just listed, and we're going to go back and add some more to it, everything we just listed is going to be located here in the cell body. That's what we're focusing on right now. The cell body, again, is going to have a nucleus, which is filled with DNA, and then we've got mitochondria. We can't see them in this picture, but we have another picture that will show the detail. The mitochondria and rough and smooth, rough ER that we mentioned is going to be found in here. We've got the dendrites that are coming off of the cell body, and then our axon. So let's back up a little and fill in a couple of other things, and then we'll look at a more detailed picture. Okay, so continuing to focus in on the cell body, we've also got what are called Nissl bodies. The Nissl bodies are just areas where the rough ER and the ribosomes are numerous. So because they're kind of clumped together and there's quite a bit of rough ER and ribosome in the cell body, this will make the neural tissue appear gray. And this is where the term gray matter comes from. So in your previous study, you may be familiar with gray and white matter. This is where we get gray matter because the dense, numerous areas of rough ER and ribosome show up dark when we look at neurons. And this makes, again, that neural tissue appear gray. The dendrites are the highly branched fine processes that come off of the cell body, and they receive information from other neurons. They make up about 80 to 90% of the neuron surface area. The axon that comes off of the cell body is very long and carries electrical signals, or as I mentioned before, action potentials, to the target, whatever the target may be. The axon structure is very important to its function. Inside the axon, there will also be cytoplasm, just like there is in the cell body, and we call it the axoplasm. Kind of makes sense. The axolemma is the cell membrane that surrounds the axon. The axon hillock is the thick section of the cell body that attaches to the initial segment of the axon. Okay, so let's take a look at what all that means. Okay, so here we've got our cell body, the main bulk of the cell, and then we've got down here the axon. So if we focus in on the cell body, we've got again our nucleus, and we can see surrounding the nucleus are dense areas of rough ER and ribosomes. And that is what we call together the Nissl body. So we can see that listed here. Nissl bodies made up of rough endoplasmic reticulum and ribosomes. And remember, these dense areas is what makes neurons look gray. So this is where we get gray matter from. All of our fine branch processes are called the dendrites. We can see mitochondria in here. We can see parts of the cytoskeleton, which are these little thin, kind of thread-like looking, very faint areas here. An example of a part of a cytoskeleton is called a neurofilament. This just gives the cell body shape and structure. Okay, and then we've got, let's see, going back, we're going to add on these terms and then go a little further with our drawing. The structures of the axon. So we just talked about how the axon comes out of the cell body, and when the axon ends, we have at the very bottom of that synapses. Some of the other things to note would be collaterals. Collaterals are branches of an axon. So sometimes an axon can branch, and it can branch a few times or many times. The telodendria, which means tree, are the fine extensions of the distal part of the axon. Remember, distal meaning distant. So the end of the axon, those are the fine extensions of the distal axon. And then the synaptic terminals, which are the tips of the telodendria. Okay, so backing up to our more kind of basic view, we've already covered the cell body pretty well. Dendrites, cell body, nucleus, axon, which ends in telodendria, okay, which means tree. You can see it looks like branches. The telodendria end in little tips known as synapses or the synaptic terminal, or where it terminates. Okay, we covered this picture. All right. So the synapse is the area where a neuron communicates with another cell. The synapse being the end of the neuron. And the synapse is where, again, the neuron can talk to another cell. And that cell could be a muscle cell, or it could be another neuron that it's attached to or close to. So at the synapse, there is a presynaptic cell. This is the one that's sending the message. And then there's a postsynaptic cell. This is the one that receives the message. And in between the two, we have a synaptic cleft, which is a gap that's going to separate presynaptic membrane from the postsynaptic membrane. So here's a view, and then I've made a drawing for you that I've incorporated into this to kind of simplify it a little bit more. So here is a presynaptic cell. So this is like one neuron. Here's the cell body and all the little dendrites going around the edges here. And here's our axon. The axon ends in synapses. And this particular neuron has 1, 2, 3 synapses. Now, this neuron is the presynaptic cell. It's the one that's sending the message or the impulse, and we can see that as listed here. So here's the impulse going down through the axon, the message, and that message is typically delivered through the synapse to another cell, which can be another neuron, which is the case in this picture, or it could be another type of cell like a muscle cell. So here is another neuron. There's its cell body, its dendrites, and its axon. So in this particular example, this presynaptic cell is attached to another neuron, and we'll call this the postsynaptic cell. It's the one that's receiving the message. Again, postsynaptic cells can be neurons, or they can be other types of cells like muscle cells, for example. So here's a close-up view that I have included to show you what it looks like at the tip of a synapse. Okay, so to give you an idea, what we're doing here is just kind of zooming in at this area right here where the synapse touches the postsynaptic cell. Alright, so here is our synapse, also known at the tip as the synaptic terminal, or where the synapse terminates. Here is our postsynaptic cell. Again, this could be a neuron or another type of cell in the body, like a muscle cell. In between the two, we have a little area, and this is called the synaptic cleft. Okay? And we'll kind of go into a little bit later what happens at the synaptic cleft, but just to give you kind of a broad look. When an impulse or message is sent through one neuron to another cell, typically right here at the synapse, we will have a release of neurotransmitter. The neurotransmitter will flood into the synaptic cleft and then bind to the postsynaptic cell to deliver a message. Okay? And the postsynaptic cell, depending on what it is, if it's another neuron, it will just continue passing that message along, or if it's a muscle cell, it may contract. So we'll leave that open-ended for now. The synapse can also be referred to as the synaptic knob, because it sort of looks a little knobby at the end. This is the expanded area of the axon of the presynaptic neuron. It typically will contain synaptic vesicles of neurotransmitters. Okay, remember that vesicles, back to our general biology, vesicles are little membranes filled with something. And they're typically released from a cell by the process of exocytosis, or ejection from the cytoplasm. Synaptic vesicles in this case are going to be filled with neurotransmitters, which again are chemical messengers. These chemical messengers will be released at the presynaptic membrane and will, like I mentioned before, bind to receptors on the postsynaptic cell membrane. Neurotransmitters, once they have been used, will be broken down by enzymes and then reassembled to be used again should another impulse come through. So here's a close-up view again of a synaptic knob. Okay, so we've got the telodendria here, which is coming off the axon, and we're ending in a synapse or synaptic knob. Okay, so this is our presynaptic neuron here, the one that's sending the message, and here's the postsynaptic cell here, which could be a neuron or some other type of cell. In between, we have the synaptic cleft, which is clearly marked. So whenever this neuron sends a message, that impulse will travel to the synaptic terminal, and we will have synaptic vesicles filled with neurotransmitters that will be released into the cleft. Those neurotransmitters will flood into the cleft and then bind to receptors on the postsynaptic cell. The binding to those receptors will continue the process by reforming an action potential that moves through the cell and either creates an action or continues propagation of the action potential, depending on what the scenario is. Neuroglia, remember, make up half the volume of the nervous system. So back at the beginning, we talked about how neurons and neuroglia are the two cell types we find in the nervous system. We just covered neurons pretty closely, so now we're going to take a look at some examples of neuroglia. Neuroglia make up half the volume of the nervous system, so they're very numerous, and there are many types of neuroglia in the central nervous system and the peripheral nervous system. And I'm going to separate these out for you so that we know which belong with which system. We're going to start with central nervous system neuroglia. So these guys are found in the central nervous system specifically. We have first ependymal cells. The ependymal cells are cells with highly branched processes. So they've got lots of little processes that come off of them, sort of like how the dendrites look. But we'll see a picture just to give you a comparison. Astrocytes are large cell bodies with many processes that come off of them as well. Oligodendrocytes are smaller cell bodies with fewer processes. And the microglia are the smallest and least numerous, with many fine branched processes. So focusing in on the ependymal cells first, we're going to find these lining the central canal of the spinal cord and ventricles of the brain. So it makes sense that they're found in the CNS, since those are the two things that belong to the CNS. They will secrete cerebrospinal fluid, which is also known as CSF. And we know that's the fluid that surrounds, cushions, and insulates the brain and spinal cord. These cells also have cilia or microvilli that will circulate the CSF. The cilia, remember, cilia are hair-like projections that are constantly moving, and they're going to make sure that CSF is constantly circulating. They will monitor the CSF and also contain stem cells for repair. Astrocytes are important because they're going to help maintain the blood-brain barrier, or the BBB. And the blood-brain barrier is important because it kind of separates the brain from general circulation. Obviously, the brain gets the blood that it needs, but not every chemical, medication can get into the brain, which is really good. It's a way to safeguard the brain. So it's going to help maintain that, to isolate the central nervous system. It will help repair damaged neural tissue. The oligodendrocytes are there to help sort of myelinate or insulate, another word for, the axons. And we'll take a look at what myelination looks like in just a minute, so kind of hold that thought. Alright, so in this picture here, we're going to focus first on the right side. So we've got up at the top a little chart to keep things organized. We've got neuroglia, which are found in the peripheral nervous system or central nervous system. Thus far, we have only talked about the central nervous system neuroglia. So up here, kind of looks a little bit like a little octopus or squid. We've got a ependymal cell, which has a cell body and some little tentacles coming off. These are the ones that line the ventricles of the brain and the spinal cord, and they're going to help to circulate and monitor the cerebrospinal fluid. We've got oligodendrocytes. These are going to wrap around the axons of the neurons to insulate them in the central nervous system. Astrocytes help maintain the blood-brain barrier. Microglia are very small with fine branches, and their job is to remove cell waste and get rid of pathogens by phagocytosis, which remember means cellular eating. Okay, so here's a picture of neuroglia in the central nervous system. Okay, So over here, just to kind of orient you, this is the central canal of the spinal cord. And surrounding it, we've got our ependymal cells. Remember, these are the ones that are going to help circulate and monitor the cerebrospinal fluid. Here, this large blue structure, this is a neuron. We can see it's the cell body, and coming off of it is the axon. Here's another neuron, and there's the axon. There, and let's see, that's the most obvious one. So we'll focus on this one for now. This guy right here is an oligodendrocyte. Notice how it is attached to a neuron. That's what this blue is, and it's wrapped around it, sort of insulating it. Okay, and that will become important a little bit later. Here we can see a microglia, which is one of those small neuroglia that have the fine branched processes. And remember, these guys are going to phagocytize pathogens. Okay, so backing up to our chart actually, on the left side, let's look at the peripheral nervous system neuroglia. We've got two, satellite cells and Schwann cells. Satellite cells are going to surround the cell body in the neuron, in the ganglia, and we'll talk about what ganglia are in just a few minutes. They're going to help regulate oxygen, carbon dioxide, and nutrient levels around the neurons in the ganglia. Schwann cells surround all axons in the peripheral nervous system. They're going to be the insulators. Now, these two are similar, but what separates them is the oligodendrocytes insulate neurons in the central nervous system, whereas the Schwann cells insulate neurons in the peripheral nervous system. So that's how we need to keep those two separated. Okay, so looking at a Schwann cell. Schwann cells are going to insulate the neuron, remember, in the peripheral nervous system, not the central. Schwann cells can work in two ways. They're found on myelinated axons and unmyelinated axons. So what the heck does that mean? This is a myelinated axon. So let's orient ourselves. We have the neuron cell body here, and we have the axon coming down this way. Okay? Now notice that this axon seems to be wrapped with something here. We can see there's a lot of these little wrappings as we come down. That's called myelination. Myelination is insulation. Just like the cords that come out of your electronic gadgets that plug into the wall, like a television cord, for example, the television cord is carrying electricity to the television, and that cord is insulated, usually with kind of a black rubbery substance. And what that does, besides keeping you from shocking yourself to death, is it keeps the electricity inside that insulation so that it's going in the right direction towards the television. That insulation helps the electricity to move faster as well. Same kind of idea here. Remember that neurons are always sending information through those axons. And these myelinated areas are helping to insulate the neuron and will help to keep that information going through in a quick manner. And we'll look at that a little bit more later. But what we've got here is a cross section through the axon. And what is causing this myelination or insulation are what are called Schwann cells. Schwann cells wrap around and insulate the axon. So here is labeled, this blue thing is the axon, and then wrapped around it, like giving it a big hug, I know that kind of sounds cheesy, but that's sort of what it looks like, is the Schwann cell. That's the Schwann cell nucleus, and its arms are wrapped around the axon, insulating it, kind of hugging it, which is what we call myelination. Okay? So that is a myelinated axon. You can see that the axon is covered with Schwann cells, so we call that myelinated because it's fully wrapped. And fully wrapped axons appear white, which is why we call it white matter. This is an unmyelinated axon. And what we can see here, this is actually a group of axons. So we've got a cell body here, and we've got some axons that are kind of being joined into this original neuron, kind of in a group, okay? And this whole group is wrapped with Schwann cells. Now in this situation, the axons are not fully wrapped. And because they are not fully wrapped, these are called unmyelinated axons. They have a little bit of insulation, but it's not nearly as extensive as the previous picture. So if we look at a cross section here, here are a collection of axons. So we've got a bunch of neurons that are kind of being held together at the axon area to form a bundle. And the Schwann cell is surrounding it, but it's not a full hug. Okay, again, I realize how cheesy that sounds. But we've got a Schwann cell here with its arms partially wrapped around the axons, but not fully. So we've got an open area here. And so we'll refer to this as an unmyelinated axon. So it's not as well insulated. Okay? So continuing our talk about the original 4 types of neuroglia in the central nervous system, we had the oligodendrocytes. And remember, the oligodendrocytes are similar to Schwann cells, but the oligodendrocytes are found only in the central nervous system. And they insulate just like the Schwann cells, which we call myelination. Myelination is important because it wraps the axon and increases the speed of the action potential to insulate the axon, so the action potential can go through quickly, and it makes the nerves appear white, which is where we get the white matter term from. White matter are areas of the central nervous system that have lots of myelinated nerves. Gray matter are the unmyelinated areas of the CNS, or central nervous system. Like that group of axons I showed you that were bundled together partially by a Schwann cell, so they weren't fully wrapped, which is why we call them unmyelinated. So here's a picture of how an oligodendrocyte works. Now remember, let's do a quick comparison. Here was how a Schwann cell did it in the peripheral nervous system, kind of hugging around, one cell sort of hugging around an area, insulating it in the peripheral nervous system. The oligodendrocyte wraps multiple times around the cell, so I've got a little cutout area for you so you can see the layering. And that's still insulation or myelination, but done by an oligodendrocyte, which again is found in the central nervous system. So 2 ways to myelinate. Microglia we talked about were the tiny little neuroglia that are found in the central nervous system that clean up cellular debris, waste products, and pathogens by phagocytosis. Okay, so ganglia. Ganglia are masses of neuron cell bodies. So where the cell bodies are numerous, we call that ganglia. Ganglia are surrounded by neuroglia and found in the peripheral nervous system. So here's a picture of ganglia. Okay, so what we've got here, here's the vertebral column. So we've got a vertebrae here, we've got a disc, and then we've got another vertebrae. Right here in the opening of the vertebrae, we can see the spinal cord peeking through. Coming out of the spinal cord are nerves. And we know nerves are bundles of neurons. So right here we can see kind of a lump. That's a ganglia, okay, or a ganglion if we're looking at it singular. So this is where a bunch of cell bodies are found, And so if the cell body is here, then this nerve must be filled with axons that are carrying information out or coming in to the spinal cord for integration and processing. Okay, so again, a ganglion or ganglia are areas where there are lots of cell bodies. Okay, so neuroglia of the peripheral nervous system, we've really already covered this using that chart. There are only two that we looked at: satellite cells, which surround the ganglia, and then we've got Schwann cells, which were the ones that insulate the neurons outside of the central nervous system in the PNS. They form myelin sheaths around the peripheral axons. One Schwann cell will sheathe one segment of axon. So we have to use a lot of Schwann cells to sheathe the entire axon, which is what we saw in that previous picture. Okay, so here is a neuron that is fully insulated by multiple Schwann cells. Okay, so each one of these things here is a Schwann cell. Okay, that's wrapped around insulating that axon. And we call these areas where the Schwann cells are internodes. The areas in between the internodes we call nodes, or more fancy, nodes of Ranvier. Okay, so either one would be acceptable. We've got the cell body, we've got the axon. Each of these is representative of a Schwann cell that insulate, also known as internodes. And then we've got between the internodes, the nodes, which are called, again, the nodes of Ranvier. Neurons perform all the communication, processing, and control functions of the nervous system. Neuroglia are going to help preserve physical and biochemical structure of the neural tissue and also help the survival and function of the neuron. So now that we know what everything looks like, we're going to take a look at how these things work to send signals. So we first have to understand what a transmembrane potential is, and this is something we do go over in general biology before we get to anatomy. And transmembrane potential, we're going to look at ion movement and electrical signals. So all plasma or cell membranes produce electrical signals by moving ions. Transmembrane potential is very important to neurons. This is how we are going to conduct impulses. So some of the things we've got to do in order to send signals through the neurons, we have to understand the resting potential and the graded potential first. So the resting potential is the transmembrane potential of a resting cell. Okay, so this is a cell that is not actively sending a signal. Okay, so that's why we call it the resting potential. The cell is just kind of resting. And in neurons, the resting potential, if we measure it, is negative 70 millivolts. So negative 70 millivolts. We measure it in millivolts, okay, like little electrical potentials. And this again is in an undisturbed cell. Graded potential is when we have a temporary change in the resting potential, and it's typically localized to one particular area, and it will eventually be carried, but is a temporary localized change caused by some type of stimulus. And then we have what we call an action potential. An action potential is an electrical impulse that is produced by the aforementioned graded potential. And it will propagate or move along the surface of the axon to the synapse. Once this potential gets to the synapse, we will probably have the release of a neurotransmitter out of the presynaptic neuron, as we kind of mentioned before. Okay? And we may have an information processing response at the postsynaptic cell. So what the heck does that mean? Well, let's get a kind of a view first, and then we'll talk about some possibilities. Okay, so what this is saying is, remember that the central nervous system is going to attempt to direct the rest of the body using the peripheral nervous system, which, remember, can also bring information in but carries activities out as well. So it does that using these neurons. Neurons are what makes up nerves. So if the brain would like for the bicep to move, then we need to send an action potential or message through the nerves to that bicep to tell it to contract. And we do that by creating action potentials. And if you watch chapter 10, when we talked about the sliding filament theory, then you already know a little bit about this. So this is the big picture. We have a neuron that is at resting potential, meaning the neuron's not really doing anything right now. Well, it's doing things, but as far as an action potential, it's not carrying out an action potential. So our resting neuron, if we measure the transmembrane potential of it, it's found naturally at -70 millivolts. Well, if we have a stimulus to try to get this neuron to fire, to direct a process, that stimulus will create a graded potential, which means a change in the transmembrane potential, typically meaning that the membrane potential will be more positive. Okay, so we might go from -70 to -60 or -50 or even higher than that. So the graded potential is caused by a stimulus, and the graded potential can produce an action potential. And we'll look at how this happens in the coming slides. The action potential fires and triggers release of a neurotransmitter. Neurotransmitters make things happen. Either the neurotransmitter will tell another neuron to fire another action potential, and we'll continue that path until we get to our target, or it will direct something to happen. Say this was a muscle cell. The neurotransmitter would direct the muscle cell to contract. So the three requirements for a transmembrane potential. We know that the transmembrane potential for a resting cell is -70. So in order to have that -70, we have to have concentration gradients of ions. So the two big ions we're going to be looking at here are sodium, or Na, and potassium, which is K. Sodium and potassium are going to be located, well, sodium's going to be located outside the cell and potassium's on the inside, and I'll show you a picture of that in just a second. The cell membrane of a neuron is selectively permeable, which means that it allows some things in and some things out, but it's pretty picky about what it lets in and out, which is a good thing. And this difference in concentration gradient between sodium and potassium is going to help us have the resting membrane potential of -70 millivolts, which is again where the neuron's found at rest. Now, in order to get the neuron to fire and something to happen, like a muscle contraction, we've got to change that resting membrane potential. We want to make it more positive. If we can do that, this will cause an action potential, and we will have movement if we're talking about a muscle cell, for example. So this picture is just to remind you of what it's like for a typical cell, what a typical cell's environment is. So this represents a typical cell. That's what the square is here. And a typical cell, typically, it's a lot of typicals, typically is surrounded by two major things that we're going to focus on. Sodium, there's a whole bunch of sodium outside the cell, and there's also some chlorine outside the cell as well. Okay? And inside the cell, we've got, chloride. Inside the cell, we've got potassium. Okay? Potassium is largely found inside the cell. So there is definitely a concentration gradient. There's more sodium outside the cell than there is inside the cell. and there's more potassium inside the cell than there is outside the cell. So the extracellular fluid of a cell is made up largely of sodium and chloride. We're not going to worry about anything else right now. And inside the cell, we've got lots of potassium, but we also have, which I did not draw in here, we have a lot of negatively charged proteins. Okay, so overall, the inside charge of a neuron is found, again, negative 70 millivolts at rest. Boy, this is a busy picture, isn't it? But I'm going to try to simplify it for you, and then I've got a drawing of my own that follows this to sort of simplify it. Okay, so let me tell you what you're looking at here first. So what we're looking at is the cell membrane of the neuron. Okay, so this thing right here, this blue, is the cell membrane. The yellow is the cytoplasm. Okay? Now inside the cytoplasm we see a few things. We've got some proteins that are negatively charged. These guys are contributing to the negative 70 millivolt measurement we've got because they're negative. We've got some kind of orangish colored circles here. And let's look over here at the key. So the orange circles represent potassium. And as we saw in the previous picture, we should have a lot of potassium inside the cell, and we do. Okay, so a lot of those orange circles. Notice we do not see a lot of potassium outside the cell, which also is to be expected. Outside the cell, we see two things. We see these green circles, which represent chloride, and we see these purple circles, which represent sodium. Okay, so notice there's a lot of sodium and chloride outside the cell, but not a lot inside the cell. Okay, and that matches what we just looked at. So a lot of potassium on the inside and a lot of sodium and chloride on the outside. Now notice as well we've got some channels here, some protein channels. Protein channels are found in cell membranes and they allow ions to move back and forth. We have some specific types of channels. We have leak channels, which are always open and typically allow leakage of sodium or potassium across the membrane. So there's always a slow leak. Then we have the active transport pumps, which require energy to work. Okay, that's why we see ATP here, which is our energy currency of the cell. So if you focus in here, you can see that this one is labeled as a potassium leak channel, and this one's labeled as a sodium leak channel. So sodium will be slowly leaking through here all the time, and potassium is slowly leaking through here all the time. Now, the leaking is caused by the difference in concentration gradient. Remember that passive transport happens when we've got more of one thing on one side of a membrane than we do on the other side. So wherever there is a higher concentration, remember concentrations will move from high to low. So we've got a lot of potassium inside the cell, so it's going to slowly leak to the outside. And we've got a lot of sodium on the outside of the cell and not very much on the inside, so it's going to constantly be leaking inward. Now to keep the sodium on the outside where it belongs and the potassium on the inside where it belongs, we have the sodium-potassium exchange pump. And this guy works using energy to try to continuously put the sodium back outside where it belongs and the potassium back inside where it belongs. So it's constantly working to sort of fix what those leak channels are allowing to happen. And this will help us to maintain the negative 70 millivolt charge in a resting cell. Now over here, we have a little probe that's been inserted into the cell that's measuring for us the millivolts. And notice right now we're clocked at negative 70 millivolts because we are in a resting cell. Now I redrew that picture, that confusing picture. I redrew it, kind of simplified a little bit to maybe make it a little easier to see. So remember we have this represents our cell body of the neuron. We've got the plasma membrane and we've got the cytoplasm. On the outside, remember, we've got a lot of sodium and a lot of chlorine. And on the inside, we've got a lot of potassium. Now remember that the potassium leak channels are always allowing potassium to leak out. And the sodium leak channels are allowing sodium to leak in, and that's again because of the difference in concentration gradient. To rectify that constant leakage, we have the sodium-potassium pump in the middle, which works by energy, by burning ATP. The sodium-potassium pump tries to keep everything where it's supposed to be to maintain that negative 70 millivolt charge. So for every 3 sodiums that the sodium-potassium pump pumps back out, 2 potassiums are pumped in. Okay, so in red I've got potassium leaking out naturally because of the concentration gradient. Sodium-potassium pump says, hey, not so fast, get back in there, and it pumps 2 potassiums back in. Meanwhile, the sodium leak channel is allowing sodium to leak in, and the sodium-potassium pump says, not so fast, 3 sodiums, get back out. So this is a constant pumping process going on all the time to try to maintain those concentration gradients and keep us at negative 70 millivolts. So the sodium-potassium exchange pump is powered by ATP and will carry 3 sodiums out for every 2 potassiums it brings in. This balance will help us to maintain our resting potential at negative 70 millivolts. The changes in transmembrane potential. Transmembrane potential, which remember is negative 70 millivolts at resting, if it changes, this will help us to have an action potential, which means we'll carry out some type of action potential to cause the release of a neurotransmitter, which can direct a change in the body. Transmembrane potential changes in response to changes in membrane permeability or results from opening or closing specific membrane channels. Membrane permeability to sodium and potassium will determine the transmembrane potential. We've already talked about this. We've got passive channels and we've got active channels. The passive channels are the ones I refer to as leak channels. They're always open and they're always leaky. So we've always got sodium leaking in and potassium leaking out. Active channels open and close in response to stimuli. The transmembrane potential exists across the membrane because the cytoplasm and the extracellular fluid have different ionic balances. And we looked at that already. There's more sodium on the outside than on the inside, more potassium on the inside than the outside. And we also mentioned that the plasma membrane is selectively permeable, which means it's picky about what comes in and goes out. So once we get to the graded potential, remember this is what's going to start potentially an action potential. At the graded potential, the opening the sodium channel will produce the graded potential. The resting membrane will be exposed to a chemical or stimulus. Sodium channels open. Now this isn't just the leaking of sodium, this is going to be a flood of sodium coming in. Sodium channels open and sodium can enter the cell. If we have a flood of sodium enter the cell quickly, again, I'm not talking about those leaky sodium channels we were talking about. These are the voltage gated channels, the ones that open if there's a change, okay, or an impulse. This will allow a lot of sodium to rush into the cell. If that happens, we have all that positivity coming into the cell quicker than the sodium-potassium pump can pump it back out, this is going to cause an action potential to happen. So this is a good thing. If the sodium channel is open and sodium enters the cell, the transmembrane potential rises, and that's called depolarization. Okay, so let me give you a definition of that. Depolarization is when we take the membrane potential or transmembrane potential from negative, which remember it's negative 70 millivolts, towards positive, or more positive towards zero, or even beyond zero. So depolarization, if we're at negative 70 and we have a rush of sodium come in, which is a bunch of positive ions, then this is going to make that negative 70 creep up towards zero. Negative 60, negative 50, negative 40, negative 30, negative 20, negative 10, negative 0, or excuse me, 0, negative 0. What am I talking about? 0, positive 10, positive 20, positive 30. So we could go as far as positive 30. So depolarization, again, when we take transmembrane potential, or membrane potential, from negative to more positive towards 0 or beyond. Okay, so here's a picture of a membrane at resting state, okay, or resting membrane potential. So we've got, again, the plasma membrane of the neuron, okay, and we've got the cytoplasm. And here are our channels, okay. These are not the leak channels, these are the chemically or voltage gated channels. These are the ones that open in response to a stimulus, okay? So remember, we have three kinds of channels we've talked about: sodium potassium pump, the leaky channels, which are the passive ones, and then we have the voltage or chemically gated channels, the ones that open only in response to a chemical or a change, a stimulus, okay? So we've got our voltage gated or chemically gated sodium channels here. Notice they're closed. Okay, and just as we saw before, we've got a lot of sodium on the outside of the neuron here. We're clocking out at negative 70 millivolts, so obviously we're at rest. But what if we don't want to be at rest? What if we want to have an action potential that will cause a neurotransmitter to be released to direct something, a muscle contraction, or to cause another neuron to fire? So depolarization is what we're going for. Depolarization is a shift in transmembrane potential toward 0 or beyond. And it's caused by a movement of sodium through the sodium channels. It will produce a current and cause further depolarization. Okay? So here's what that's going to look like. So here again is our plasma membrane and our cytoplasm. The membrane is exposed to a chemical or impulse that opens the sodium channels. Sodium channels open, sodium floods into the cell. Look at all this positivity inside of the neuron. All that positivity comes into the neuron. This is going to make the transmembrane or membrane potential more positive, right? So it's now being clocked at negative 65. So we are starting to creep up towards 0. Now this is going to spread, which we want it to do, because remember, we want this action potential to spread through the whole entire axon and make it down to the synapses. So this shows us how it spreads. The spread of sodium inside the plasma membrane produces a local current that will further depolarize adjacent or neighboring portions of the plasma membrane. Okay, so here's where it originally happened. The sodium channels open as caused by a stimulus. Sodium floods in. Okay, this causes the membrane potential to become more positive, which we call depolarization. The current, the local current that forms, causes an adjacent part of the membrane to depolarize as well. So now we've got more sodium coming in. We're creeping up towards zero here as well, negative 65. This local current will cause another adjacent portion of the cell to go through depolarization as well. So it's like a domino effect. It's going to keep continuing down the axon. We're going to have further and further depolarization down the axon towards the synapses. If we take the stimulus away, the transmembrane potential will return to normal. That's called repolarization. So to take the membrane potential from positive or zero back towards negative or its original resting place. So to repolarize, we would be returning to negative 70 millivolts. Hyperpolarization is when we increase the negativity of the resting potential past negative 70. So if we made it negative 90, for example, this creates an opposite effect. This is when the potassium channels open and potassium floods out. Okay? So hyperpolarization means we make the inside of the cell more negative. Okay? So the effects of a graded potential at the cell dendrites or cell bodies, this can trigger specific cell functions. So for example, if an axon carries an impulse down through the synapses and the synapse is attached to another neuron, this can cause that neuron to trigger specific cell functions. Exocytosis of a gland, for example. If the axon and synapse of the neuron that is firing is attached to a muscle cell, it will cause the muscle cell to contract. Okay? So again, here we have at negative 70 millivolts, we're at resting state. So we're kind of backing up and going through this again. So we're at resting state, negative 70 millivolts. These positives represent potassium, and the purple positives here represent sodium. And notice that both of the channels are closed, so there's no movement of ions yet. We're at rest, negative 70. Okay? So 4 steps in the generation of an action potential. So we're going back, okay? Step 1, depolarization to threshold. Threshold's kind of a sweet spot. It's a magic area. We want to get to threshold. If we get to threshold, contraction is imminent. It will happen. Or the action potential is imminent. It will happen. Okay? So threshold is our checkpoint. Step 2, we want to activate the sodium channels so that depolarization can happen. Remember, that's when sodium rushes into the cytoplasm and makes the inside of the cell more positive or closer to zero. So we go from negative 70 maybe to negative 65 or negative 60, closer to zero, from more negative towards positive. So here's a picture of depolarization happening. Okay, so depolarization to threshold. So we've had some sodium rush in, and this local current that is now in the cell causes the millivolt number to be more positive, which is what's supposed to happen when we have depolarization. Okay. Now we want to fully depolarize. Okay, we call that rapid depolarization. So remember, depolarization getting closer to zero or even beyond. So rapid depolarization is when the sodium channels open and sodium is allowed to flood in, and we see it flooding in here. That flooding of sodium is called rapid depolarization, and all that positivity flooding into the cell causes the membrane or transmembrane potential to be clocked out at positive 10. So we've really dramatically changed the environment of the neuron, and this will cause the action potential to continue. Okay? So after we've had rapid depolarization, we want to resume to normal. What's going to happen is the sodium channels will close and the potassium channels will open. So once we get up to about positive 30 after depolarization, sodium channels will close and potassium channels will open. And as the potassium floods out, this will cause repolarization. So repolarization going back towards negative in our membrane or transmembrane potential. So going back to checkpoint, negative 70 millivolts. Okay? So this is a picture of where we start repolarization. So we've made it all the way in our depolarization. We've allowed so much sodium to come in that we're now at positive 30 millivolts. Once we get to positive 30 millivolts, sodium channel closes. As you can see, it's done here. Potassium channel opens. Potassium is allowed to leave. And when potassium leaves, that positivity leaves, it helps return us back to negative 70 millivolts, which is our resting position. So when the sodium channels close, the membrane will reach normal resting potential, about negative 70 millivolts. When the channels are finished closing, typically we overshoot a little. So we often go past negative 70 as we're repolarizing and may end up at negative 90. This is called hyperpolarization. Now, whenever we are hyperpolarized, we call this the refractory period. And the refractory period is a period of time where the neuron cannot be stimulated. Sort of a short resting period. Okay? Now, in this picture, we can see the return to normal permeability. So this shows hyperpolarization as we get ready to get back to our normal permeability. Okay, so in this picture we've got more negative inside than we usually do, so we've overshot. This is when we allow a lot of potassium to leave, and we typically go past negative 70, which we call it the refractory period. Remember, the period of time where we cannot be stimulated, the neuron cannot be stimulated. So that's why we're clocked out there at negative 90 millivolts. So as I just mentioned, the refractory period, this is the period of time that the neuron membrane will not respond to additional stimuli. This is the resting state after an action potential. All right, and in this picture here, we can see a chart kind of clocking our progress, okay? So this chart shows the membrane potential on the left and the time on the bottom. So on the left, we have negative 70, which is where we start. We've got negative 60, which is our threshold, that's our magic spot. Negative 40, 0, and positive 30, okay? So we started at negative 70. Our neuron resting membrane potential started at negative 70. We depolarized, remember depolarization, going towards positive, or becoming more positive, and we did this because we allowed sodium to come in to the cell. So depolarization, we go up, 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 up until we get to positive 30. When we got to positive 30, the sodium channels closed, and when the sodium channels closed, this allowed, we then had the potassium channels open, so potassium or positivity floods out. So this caused us to drop or repolarize, and we overshoot a little bit, so we ended up around negative 90, and this was our refractory period, a period of time that we cannot be stimulated in this neuron. And after the refractory period, we're back to negative 70, ready to have another fire of the neuron or action potential. So how does the action potential go all the way down the axon? How do we keep it moving forward? Now remember, I'm going back a little bit here just to show you sorry guys, I went back too far. Let me go back to show you the original picture of that neuron. Okay, so we're going to talk about two types of propagation of the neuron, but in order to do that, I think it would be helpful to see that visual one more time of the overview of what we're trying to do here. Here we go. All right. So here's what I was looking for. So we want to have an action potential or fire that will spread through the axon to the postsynaptic cell. The postsynaptic cell is the cell we want to affect. And it could be a muscle cell or it could be another neuron. So the idea is to get the action potential, which is all that depolarization, repolarization stuff, to flow down the axon to the synapses where a neurotransmitter can be released to cause some type of action. Okay? And there are two ways to carry the action potential down the axon. And those are known as propagation. And there's two ways to propagate. Get back to our original position. Okay. So propagation of an action potential. Propagation is to move an action potential along the axon. Okay, we want to go along the entire length of the axon. And it's a series of repeated actions. It's not passive. There are two methods of this. We have continuous propagation and we have saltatory propagation. Continuous propagation happens in unmyelinated axons, and saltatory propagation happens in myelinated axons. Now remember that unmyelinated means they're not fully insulated, and saltatory propagation means that they are fully they're in myelinated axons which are fully insulated. Okay, and we talked about the Schwann cells and the oligodendrocytes, so hopefully that's familiar. All right, so continuous propagation. Continuous propagation of action potentials along an unmyelinated axon. This affects one segment of the axon at a time, and because it's not fully myelinated,